This protocol introduces a defined feeder cell-free method for differentiating corneal limbal epithelial stem cells from human pluripotent stem cells.
Limbal epithelial stem cells are responsible for renewing the corneal epithelium in the healthy eye. Damage to these cells leads to a condition known as limbal stem cell deficiency and to the loss of corneal clarity.
The cell culture methods shown here enable efficient production of limbal epithelial stem cells for future corneal cell replacement therapies.
To begin, passage and maintain the feeder-free human pluripotent stem cell culture as outlined in the text protocol. Make sure to use only high-quality human pluripotent stem cells as starting material for differentiations. When ready to induce embryoid body formation, warm all of the needed materials and reagents to room temperature in a laminar flow hood. Detach the feeder-free human pluripotent stem cells to suspension by adding 500 microliters of xeno-free trypsin EDTA to each well, and incubating at 37 degrees Celsius with 5% CO2. After 3 minutes, remove the cells from the incubator and check the cell morphology to determine the optimal phase for trypsin removal. Carefully observe the cells to determine the optimal time to remove the xeno-free trypsin EDTA when the cells have rounded up but have not detached completely. At the optimal time, remove the xeno-free trypsin EDTA from the cells. Add defined trypsin inhibitor and gently pipette to detach the cells. Collect the single-cell suspension in a 50 milliliter centrifuge tube. Centrifuge at 300